All right, so our next speaker is Dr. Loren Kirschner, who's going to talk about the neurological effects of acetogenins and what do we really know.
There's the clicker. No pointer.
The big one is forward and then the small one's back.
Okay. Hey, you know what's really nice is Kirk got like half of my stuff, so I have about a minute's worth of things. I'll cram it into 15. How's that? Yeah. So I got just a few questions to start out with. First off, I'm Dr. Loren Kirschner. I'm an internal medicine physician, a critical care physician. Was an old Army doc for many years as well, and also a pawpaw grower. So I've been involved in pawpaws for about 25 years. So I have had kind of the intimate experience of reading all that literature as it's come out over the years, and it was always interesting to me. But the fact that I'm still here is, I think, is very helpful. I haven't had that much of a concern, but I wonder, prior to today, who has had any concerns about this issue of any significance to the point where you might make some decision making about whether you grew them or not? Hold them up. I saw a few at least. So this issue to me is one of those that I'm an old Army guy for years. My brother was a Navy guy. I'm going to point him out. And I look at these things from a Navy standpoint as a torpedo under the water sometimes. So I'm looking at it from the future of the industry, and I think while I don't have any specific concerns about it, and the FDA I know has issued that statement, in the past, and the nature of the development of this literature base has really died off. Okay, but still, in order to move an industry forward, I just look at this as one of those that it's like a torpedo under the water. It doesn't care, but at some point it's going to hit the ship. Right? And it doesn't matter where it came from, it still causes the same, it has that potential to cause the same destruction. I think that if folks were interested in developing, higher quantities of fruit, or eventually doing, more mass use of fruit, this could potentially become another issue. Recently, Dan, back here someplace, forwarded me a little video clip on YouTube about this once again. Somebody resurrected, it was a physician who wrote an article. I looked at it. It was interesting. I've read all that literature before, but it's like these little torpedo, them little ones out there. Anyone see that, by the way? It was a YouTube, not recently, pretty recently. Pardon? Yeah, he's like a nutrition sort of person and all that type of stuff. So you're really looking at this. I'm going to take this just a little bit different direction. I want to highlight towards the end of it, actually, where I think a lot of the problem is, actually, if there is an issue at all with this, with acetogenins. So let me see what I can do here. And if it doesn't interest you too much, you can at least look at our pollinators. So they got wove in there, okay? So I'm going to hit some of those, couple more points on the brief history of this, and a very brief look at acetogenins, and a whole bunch of black boxes, if you will, about where the problems actually might be, if there are any, once again. So again, you got to look back to the 90s to this. It's actually earlier than that, even, when people recognized that they're really kind of just broadening this out. Worldwide, there were a number of clusters of Parkinson's, and particularly in islands in the Pacific, but also in Guadalupe, that were identified. And there was about 11 of them, of which publications came out of, about concerns, first off, about clusters of this atypical Parkinson's type of syndromes, okay? And focusing in specifically about the Guadalupe experience, they tried to make some of the correlations, and I have to give them folks credit for at least looking at this, right? Because it was of concern. So, these are what I refer to as, in a sense, epidemiological types of studies. These were case control types of things where people were looking at, so many cases of atypical Parkinson's and matching them up with a control person, a control patient who doesn't have the symptoms, and then seeing what they were doing, if you will, that might impact why they have a problem. So the case control types of studies, they are great for hypothesis generation. They're not great for making any distinctions or true correlations. They're correlations but not causation, if you will, right? Correlation but not causation. So you're looking to, but at least generate hypotheses about what you might want to look at. So acetogenins came up. Oops, I went back. There we go. So, and particularly at the bottom here, because a lot of these clusters that they were looking at didn't have clear genetic causes. We know that in terms of Parkinson's, there's actually some genetic basis to this, at least in a number of cases. PSP, which is called progressive supranuclear palsy, and that's what we're referring to as the atypical Parkinson's. Okay, it has some features of Parkinson's, but it's not. And I kind of hit a few of the the symptomatology of it on the next slide here. But they were looking at, because there was no clear genetic basis, what might in the environment be involved here. So busy, and I'm going to skip through some of this, but the atypical Parkinson's is the second sort of box there. That's kind of the distinction. So you got these clinicians who are trying to figure out, based on somebody walking in with a certain neurologic problem, trying to decipher, is this person got Parkinson's? Is this kind of an atypical Parkinson's? They're all today referred as neurodegenerative conditions. It means that certain parts of the brain are actually deteriorating. Alzheimer's, the most common form of dementia in the world, is a neurodegenerative condition. A lot of this depends on what parts of the brain are actually being affected by the neurodegeneration, right? So this is no different from that in a sense. But they found those high numbers of PSP patients in Guadalupe and started having concerns about that. If one wants to look at those original studies that are available out in the literature, and actually they're free, so you could look on, websites at those and read those original papers, the case control studies. It's kind of backed up a little bit initially with some ananascent experiments where they're injecting rats and kind of, Kirk was talking about some of that stuff. And it kind of confirmed, that they were actually developing pathology in the brain that was resembling PSP. So that was an important key component. What is referred to for PSP as, quote, tau pathology, tau protein. It's a specific protein in the brain, in certain parts of the brain, neurons. So it was being affected. So that's kind of what kind of propelled that forward. Acetogenins, I'm going to skip mostly through because Kirk hit most of that information. I find that there's actually maybe a lot higher numbers, but there's also been a lot of synthetic acetogenins released, people synthesizing, trying to look at, from a cancer standpoint, more toxic type of acetogenins, even above and beyond what you find in nature. What's interesting to me is, so structurally, as Kirk pointed out, or Dr. Pomper pointed out, they're moderately large, they're lipophilic compounds, they're fat compounds, they're fat derivatives, and that plays a big part in pharmacology and pharmacokinetics. Is any pharmacology people? Any? No, good. Then I can say what I want. No, I'm just kidding. So pharmacology is very important here, but these are moderately large in their lipophilic compounds. That lipophilic means that they're going to have an easier time penetrating fat layers, okay? And that's particularly important because every cell in everybody's body here, every cell that they have has a fat component, right? The cell membranes are fat components, and so you get those compounds through lipophilic if they're lipophilic. So they're easier to get into cells, okay? And that's important. But it also has some other things about it we'll talk about next. Acetogenins, once again, they have been shown to be this mitochondrial respiratory chain, one component, the first component of that. And what happens if you have a mitochondrial complex 1 inhibitor, it basically binds either reversibly or irreversibly to that, and it grinds the production of what is referred to as ATP to a halt. ATP is the energy currency of our cells, okay? It's the currency, if you will, of the energy movement throughout cells. If you grind that to a halt, the cells die. Simple, okay? Now, interestingly enough, somebody doesn't have to admit to this, but when I think about drugs, and I was bringing this up the curve, who on here, somebody in here, statistically speaking, should be on metformin? Was? Anybody else willing to, anyone heard of metformin? For diabetes? Okay. Well, metformin is a complex 1 inhibitor, by the way. And there's other drugs that are that way. And a lot of other compounds, and rotenone, and paraquat, and diquat, and a whole hodgepodge of other ones out there in the world. Did they all cause, problems? Not necessarily. So, but this was the initial linkage. They wanted to make that connection potentially because it made some sense logically that might be the ultimate reason why the cells were dying. So, and leading to this tau pathology. So, and again, if we know in that next slide there, if you do this in a petri dish, so to speak, acetogenins will cause cells in cell culture to develop, neurons specifically in cell culture to develop tau pathology. So there seems to be some linkage that way as well. So, but again, other compounds, lots of them out there. A lot of compounds in the world do this, inhibit. Some are reversible, some are irreversible inhibitors, et cetera. Okay, so that's acetogenins. Now, just taking we know that acetogenins in test tubes, in petri dishes, can kill neurons. It can cause tau pathology. Where do we go from here? So there's some giant black boxes here, right? And that is called, okay, put something into your mouth and have it go into your gut, and what happens to it? Lots of stuff goes on there, right? It is not as simple as saying, I put it into my gut and I'm going to absorb it. Dr. Pomper pointed out that it's actually a fairly low rate of absorption through the GI tract, and actually enter into the bloodstream, okay? That's important because in order to get to the brain, if it's going to cause brain pathology, it has to go through a lot of different steps here. So I'm going to take you a brief little walk through some of the pharmacology, if you will, of what happens here. So first off, you would classify these compounds as what I call class 4. I think this will come up. There we go. Class 4. On a classification scale, this is important because class 1, which I now blocked, they have a high solubility and a high permeability, which means permeable means I can get it through the cell membranes. High solubility means it's going to dissolve in water, aqueous solutions, and that's very important to get drugs into your body. So a lot of this is always a nightmare for developing new drugs because you have to figure out how to get it into the cells you want it to get it to, right, ultimately, without it being broken down. So class 4 is referred to as low solubility, low permeability. The low solubility is because it's a fat type of substance, so it won't go into water well. Low permeability is a size factor. Acetogenins are fairly large molecules, and they're not going to penetrate cells very well. So that's a first barrier. It's very likely that these things, why it accounts for that 3.2% getting into the bloodstream, that's into the bloodstream part. That's part of it. So you put that much, if you ate your 30 pawpaws today and you got to absorb all of that and you get just a low amount that actually makes it through the GI tract into the bloodstream ultimately, a lot of it's due to that. Got to get through those first hurdles. If you go a little bit busy slide up here, but then you got another step. So if you look all the way to the left, just kind of a diagram of the gut, but a blow up in the center part of that where it says gut microbiome or microbes. That's the representative of the gut wall. You got to get this through the gut wall. You got to get it into what we call the portal system. It's going to go from there to the liver. And then the liver is known as our what? Great detoxifier, right? It has great capacity to detoxify a lot of different substances. That one, to my knowledge, I'll read it back to myself here. To my knowledge, there are no major studies that have documented acetogenins and how they're metabolized in the liver. None. Do you know of any, Dr. Pomper? I know of nothing. That's a big issue, okay? We don't know what the liver does with them, and the liver is our one of our major detoxifiers, so we don't know that. The next part of that, systemically, it's before that, is we don't know what the pH in the stomach does to these things at all. This is another big black box. So a lot of getting drugs into the system, you got to get it through the GI tract, right? You got to get it through the acid environment of the stomach. Then it's got to get into that portal system. It's got to get through the liver before it gets to the bloodstream. And then, after all that, you got to get it through something called the blood-brain barrier. We have something, and all other animals do, you have something called the blood-brain barrier. It is going to block most things from getting into your brain. That's crucial, okay? And it's a big step, it's a big hurdle. So anyone who does any drug research looking at sort of, quote, brain active compounds knows this. It's a very difficult industry to work in because you got to get through the blood-brain barrier. The brain is very, very particular about what's going to get in. If you would try to predict this, blood-brain barrier diffusion is possible based on what we call that lipophilicity, where it could go through fats. However, the size is very limiting. Usually anything less than what we call 400 Daltons is not going to make it in. Acetogenins are basically 4 to about 600 Daltons. They're typically larger than what you would expect to actually get into the brain. To my knowledge, there's nothing published about getting drugs or getting acetogenins at all across the blood-brain barrier. So while you can put acetogenins in a petri dish, that is not physiology. That's not, right? That's not how the human body works. So there's other things going on, and that's actually what's much more interesting here. So at least most people in here, hopefully, are at least somewhat knowledgeable right now about the gut microbiome. Anybody interested in that? Anybody hear of it? Anybody using probiotics that are enhancing that maybe, or that type of thing? So the gut bacteria and the gut microbiome is a key part of this, this whole process. So you put something into your gut, it is not sterile, right? It is not sterile in there. And there's a ton of things goes on with whatever you eat, whatever you consume, whatever you put in here, the interaction between the gut bacteria and what's going to be some type of outcome, for whatever you put in there. If you skipping across all those other things, that's some of the things that the gut microbiome do. But it's a big one about detoxification and metabolism of what are referred to as xenobiotics. These are compounds. Xenobiotic would be something that's not naturally inside of me. Acetogenins would not naturally be inside of me. We would call this a xenobiotic that way. So there's a lot of detoxification things that goes into there, that happens in the stomach. Okay? Now, if you look at this from a different perspective for just a second, and this is the most interesting piece, and I got like a couple of slides, and then I'll be finished up here. Anyone heard of the term gut dysbiosis at this point in time? Dysbiosis. So dysbiosis would mean when we're looking at the patterns of the types of bacteria in the gut, we all have some very peculiar patterns to us. There's minor differences, but what is considered a healthy microbiome being a standard, dysbiosis is something other than that. If you had an antibiotic recently, you would at least transiently have a dysbiosis. You can't avoid it. I can't avoid that, right? It happens. So in other words, I put an antibiotic into my gut that selectively kills off certain strains of bacteria. I've got a dysbiosis going on, at least temporarily. And eventually, it tends to rectify themselves. It just depends. Anybody heard of C. difficile diarrhea? C. diff diarrhea. And anybody had it yet? So antibiotic-associated diarrhea, C. diff, is a huge problem in the medical community, where somebody's received an antibiotic, and next thing you know, they had an overgrowth of a certain bacteria in the gut called C. difficile, and it causes some major havoc and ultimately life-threatening. That's a form of a dysbiosis. It happens on a much minor scale, but it still happens. But dysbiosis leads to some very interesting things called the leak. Particularly what's interesting here is the leaky gut. This is an issue where you break down the integrity of the gut wall. And this makes all the difference, because with Parkinson's, there's a lot of studies right now that have clearly shown that virtually everybody with Parkinson's disease has a gut dysbiosis. It's an issue in most people with Alzheimer's, a gut dysbiosis. And there's a lot of other problems that are associated with this. And what it does, when you stop and you think about this, what's probably going on, I can't prove this yet, okay? And I don't know whether we'll ever get to be able to prove this with the acetogenins in particularly, but if you break down and you open up the gut barrier against these types of potential toxins, that's where you potentially could be getting significant amounts of acetogenins into the bloodstream. Again, we don't know what the liver does with them. We don't know, but that's not associated necessarily with gut dysbiosis. But the finding of Parkinson's in having the majority of folks with Parkinson's having gut dysbiosis, that's a critical piece. And why do I say that is because it's very interesting. And it's been, again, it's also been shown in PSP as well to have the dysbiosis issue. But if you look at, there's a few very interesting studies, particularly one where in an animal model that we use for developing, so we can use that looks like PSP, looks like, in a sense, Parkinson's. It's called the MPTP model. This is a mitochondrial toxin, the complex 1 toxin as well. And it has been very highly linked to PSP and Parkinson's development. It's just a research type of compound now. But if you induce Parkinson's disease in animals and then induce a dysbiosis, one, you worsen their symptoms. Again, a dysbiosis meaning with an antibiotic, I screw up their gut barrier, they worsen their Parkinson's symptoms. If you then fix that, then you reverse it back. Now, if you go a different direction and you use what we call germ-free animals, then you do something very interesting there. So germ-free animals have no gut bacteria. They have to be maintained in a cage in a very specific manner. They can have no contact with outside world. They're maintaining a totally sterile gut all their life, okay? But if you induce Parkinson's in those animals and then, in a sense, colonize them with gut bacteria, even though that you set them up for having Parkinson's, you genetically breed them to have Parkinson's. If they're in a germ-free environment, they have no symptoms. If you put gut bacteria into them and cause a dysbiosis, in a sense, then you get Parkinson's to show up, which is really kind of putting all this stuff together. It's kind of like that is probably what's going on with a lot of this. I think that ultimately we know that we're moving forward as far as Parkinson's goes, and actually Parkinson's treatments and things like that, by using fecal microbial transplant, for instance, to actually reverse Parkinson's disease if you catch it early enough. It's actually effective at doing that. So there's a number of studies, a number of trials that actually have shown that. Again, my sense of this right now is that probably the biggest issue is it's a fecal microbiome issue, a dysbiosis issue. Then you come along with a toxic substance, And that probably is what, with that dysbiosis, you're allowing that toxins to get into the bloodstream and allowing the disease to develop ultimately. This would also explain why, because in Guadalupe, an extensive number of people eat soursop or in cherimoya and those type of things, and they eat all these ananaceous foods. If you look at the original study, and somebody pointed out that somebody raised their hand who actually read it. Who was it? Oh, back here. And it's very interesting because if you look back in those original studies, they had the case and the control, and about 2/3 of their controls actually consumed about the same amount of same amount of ananaceous acetogenins as the study group did. So why didn't they get sick? I don't know. So it's a hypothetical. Does that make sense? Right? Hopefully it made sense. The argument there. In other words, a large number of the people who were included in those studies who did not have Parkinson's or the controls, they actually ate as much ananaceous acetogenins as the people who actually had PSP that they were studying. So it's very interesting. I think that this is ultimately where this is going to go. I would look at this as saying, I'm going to skip forward. I'm going to skip to the conclusion here. My take on this is that, though, from a standpoint of what do we do with this in the long run, I would suggest that if it's possible, and I don't know where this comes from, because it takes money to do this type of work and look at this. I know the horticulture industry, it's not their duty in a sense necessarily. It's not their expertise. But somewhere along the way, someone needs to, I think, take this up and look at the acetogenins in terms of the basic pharmacology, answer the questions, and then move on. Otherwise, I think, and it's interesting, I got one last thing I was going to ask, and that's more of the European folks who were here, if they had any opinion about this from a European perspective, if they're aware of this problem, but also what their thoughts might be, whether this should be an answered thing. Should we be actually looking at this? Should we do the pharmacology? Should we do that type of work and answer this? Settle the question before trying to move the industry forward. I think it is a torpedo sitting under the surface, not to belittle that or anything like that. No, I think it's a significant issue potentially that we need to answer. But I think it's going to be through some of these metabolomic studies that we can do now that can be actually relatively not as costly as it would have been a few years ago. So if someone's interested in ever taking that type of work up in the university setting. And I think that's all I had. That's us. Okie doke.
So we'll take one question if there's anybody who's got tons of questions here.
Thank you, Dr. Kirsch. Thank you, Kurt. I've been president for quite a few years here. It takes a lot of time putting things together, and this hits my mailbox constantly. And I reach out to Loren on this. It took 3 years for us to present this. And I had said earlier at this podium when it started, there's a lot of professionals here that will be a lot better than for me to explain. But I also said to look around. You are our future. Please pass this on, and thank you.
Go ahead.
One more?
Yeah.
Okay. So as a fat-soluble, alcohol-soluble molecule, how does the integration into products like brandy or ice cream, how do you think that affects dosage and solubility?
The only thing I can say about that is I had some Jephthah Creek last year, and it was very good. Just a little sip of the Jephthah Creek brandy. I don't know. It's a very good question. I don't know enough about that side of the industry, so to speak. Yeah, as a lipid soluble substance.
I think when you distill it, right, it's going to be distilled. There won't be any, probably nothing left at that point. With the wine, it's hard to say, right? But if you distilled it, it's not going to be anything there.
I suppose it's going to go off in a lot of it will go out in the distillation process, I would think. But it's
I-
going to taste good.
I just wanted to say, so basically the conclusion is that we're far more susceptible to be overdosing on brandy than the acetogenins.
Probably. Yeah. I would say keep your consumption down to like maybe 30 fruit per day.
I had a little comment on the Parkinson's thing too, is our brains are 60% fat by weight and 40% is water, salt, and a couple other minerals. And so 60% is fat, but the majority of it's cholesterol and saturated fat. So when the doctor tells you that your cholesterol is bad, he's misinformed, and you should not take statins because that you need the cholesterol. Cholesterol is good for you. Your cell walls are made out of it. Everything is perfect with a lot of fat. So eat butter and papa, and you'll be good.